Hello everyone, welcome to Pathomed. Today I would like to discuss about SEZARY syndrome. The SEZARY syndrome is a cutaneous T-cell lymphoma. Also, the mycosis fungoides is also cutaneous T-cell lymphoma, but the difference is SEZARY syndrome is the leukemic variant of mycosis fungoides. That means mycosis fungoides is a cutaneous T-cell lymphoma limited only to the skin. It is confined only to the skin, whereas Sézary syndrome is the leukemic variant. Leukemic variant means the atypical lymphocytes, the neoplastic lymphocytes, they are found in the peripheral blood as well. So, Sézary syndrome involves skin, peripheral blood, also lymph nodes can also get involved. And the main characteristic differences could be mycosis fungoides is it has characteristic Poutrious microabscess. Poutrier microabscess. So this is a skin tissue bit. Here you can see this is the epidermis. This layer is the epidermal layer. Within the epidermis are the vesicles contain lymphocytic infiltrate. So this is a mycosis fungoides case showing you intraepidermal lymphocytic infiltrates, vesicles of lymphocytic infiltrate. This is called the Poutrier microabscess, which is the characteristic, the hallmark feature of mycosis fungoides. And this Poutrier microabscess mostly, almost is absent in Sézary syndrome. So, Poutrier microabscess absent in Sézary syndrome. Coming to the histopath of this Sézary syndrome, we received this skin tissue bit. This is a skin tissue bit. On low power view, you can appreciate the epidermal layer, stratified squamous keratinized epithelium. So, beneath the epithelium is the dermis. This is the superficial dermis. This is the superficial dermal layer. In the superficial dermal layer, you can visualize a lymphoid infiltrate, an infiltrate containing lymphocytes. So this lymphoid infiltrate, it is confined to the superficial dermis. The deep dermis, it remains uninvolved. And you can also see the cells, this lymphoid infiltrate, it is moving towards the epidermis, like it is going towards the epidermis. And almost the gap, the bridge between the epidermis and the dermis is very thin gap between the dermis and epidermis. This characteristic, this phenomenon is called epidermotropism. Epidermotropism. That means the lymphoid infiltrate, it shows affinity for moving towards the epidermis. The lymphoid infiltrate almost moving towards the epidermis, the dermal, the superficial dermal infiltrate of lymphocytes, they move towards the epidermis. And almost there is a gap between the lymphoid infiltrate and the epidermis. This feature, this phenomenon is called epidermotropism, characteristic of Sézary syndrome. Also, here the high power view showing you this epidermal layer with the rete. This is the papillary dermis, superficial dermis. Here there's a lymphoid infiltrate. These lymphoid infiltrate, each individual lymphocyte is atypical lymphocyte. Like the normal lymphocyte is it has a dark condensed nuclei. Here, in few of the cells, you can see the nucleus is irregular and it more or less open chromatin. That's loosely stained, open chromatin. This is an atypical lymphocyte. Atypical lymphocytes. All these are heavily dense infiltrate of lymphocytes, every cell. And it shows irregular nuclear contours and almost open chromatin in a few of the cells. This is an atypical lymphocytic infiltrate. Also here, you can appreciate the atypical lymphocytes with cerebriform nucleus. Cerebriform means the surface of nucleus has gyri and sulci like that of a cerebrum. So here, this membrane, cerebriform nucleus. Cerebriform nucleus is highly characteristic of Sézary syndrome. So this is the IHC staining. Since it is a T cell lymphoma, the T cell markers being CD1 to 8, CD28, these are characteristic of T cell markers. Here, This is a CD3 plus. It is a CD3 positivity. This is CD3 stain showing positive cells of atypical lymphocyte. These are the atypical lymphocytes. Here you can appreciate very well the cerebriform surface. The cerebriform surface. Irregular nuclear membrane. Irregular nuclear membrane with cerebriform nucleus. Almost looks like gyri and sulci of the brain. It is a CD3 positive cells. As we have said, this is a leukemic variant. That means the SEZARY cells are found in peripheral blood also. This image shows you the peripheral blood picture, and this is the atypical lymphocyte. Atypical lymphocyte. The nucleus is cerebriform, like gyri, sulci of the brain. Cerebriform here. SEZARY cell. This is a SEZARY cell. SEZARY cell in the peripheral blood. And when it gets involved, when the lymph node gets involved, the normal architecture of the lymph node, that is the germinal centers, the lymphoid follicles are absent. The normal, if there is the normal architecture of the lymph node is lost, and entirely dense infiltrate of lymphocytes are seen. So you can't find any lymphoid follicles with germinal centers. Entirely, it is occupied by this infiltrate of lymphocytes, atypical lymphocytes. Thank you, everyone. Please like, share, and subscribe. If you like this video, please like, share, and subscribe to our channel. Thank you, everyone.